If you work anywhere near biotech, AI, drug discovery, you get this feeling of just information overload.
Oh, absolutely. It's constant.
But the real problem, I think, isn't always having too much data. It's the cost of getting the right data.
Exactly. The critical stuff, the molecular insights, that's the bottleneck.
And that's a huge roadblock in research, isn't it?
It is. For decades, pathologists have had this fundamental dilemma. We have millions and millions of these standard tissue slides.
The ones stained with hematoxylin and eosin, the H&E slides.
Right. They're cheap. They're everywhere.
And they're kind of like the satellite images of a city. They show you the geography, the physical shapes of the cells, the morphology.
A perfect analogy. They cost, what, maybe $5 a slide? They're the bedrock of pathology.
But the satellite photo isn't enough, is it? To really understand cancer, you need to be on the ground.
You need street-level intelligence. You need to know which proteins are active, where they are, who they're talking to. And that kind of data comes from methods like multiplex immunofluorescence, MIF.
Which is brutally expensive.
And slow, brutally. You're talking upwards of $1,000 per slide, sometimes way more. And it takes days of really complex lab work.
So researchers are forced to just ration their samples. They can't run the tests they want to run.
Which limits studies to tiny cohorts. You miss so much.
Yeah.
So you have these two worlds, cheap, abundant satellite photos and incredibly scarce, priceless street-level photos.
And that's what we're going to talk about today, this new tool, GigaTIME, which feels like it's a direct solution to that exact problem.
It really is. It was developed by researchers at Microsoft, Providence, and the University of Washington, and its core thesis is, what if we could generate that scarce data virtually?
So taking the cheap H&E slides and turning them into the rich protein data.
Exactly. It's a multimodal AI that translates those $5 slides into, well, virtual myth images, predicting activity for 21 different proteins.
So the implication there is you can suddenly create a virtual population. You can take thousands of old archived slides
and computationally give them these incredibly detailed molecular profiles, a data set that was just physically and financially impossible to create before.
Instead of a study with, say, 200 patients.
You're now working with 14,000 instantly. It lets you fail faster, see patterns you would have missed, and, hopefully succeed sooner, all without that massive upfront cost.
That's a huge shift. So, okay, let's peel back the layers. What exactly is GigaTIME if we strip away the industry hype?
At its heart, it's an AI translator, but it's a very specific kind. We think of generative AI making, fantasy images or writing text.
Right.
GigaTIME is generative, but it's totally grounded in biological reality. It's not hallucinating fantasy. It's predicting proteomics data.
So the input is that simple purple and pink H&E slide showing the cell's physical shape.
It's morphology, yeah.
And the output is this incredibly detailed map of protein activity. What kind of proteins are we talking about?
We're talking about the heavy hitters, the ones that are critical for understanding the immune response to cancer, things like PD-L1, CD8, Ki-67.
The markers that tell you if an immunotherapy drug might actually work.
Precisely. So if real MIF is the gold standard for understanding that whole tumor microenvironment, GigaTIME is like a high-fidelity virtual simulation of that gold standard.
It just bypasses the physical lab work.
It bypasses the bottleneck. It uses deep learning to guess what the MIF stain would have looked like if you'd spent the time and the money. It effectively turns a standard pathology lab into a virtual proteomics facility.
Which brings up the big question, right? How can a simple image of physical structure predict something as complex as chemical protein expression? It feels a little like magic.
Well, it means that the H&E image has way more information in it than a human can see. The AI works on this basic principle that form follows function.
So the shape of a cell, its texture, how it clusters with its neighbors.
All of that is statistically correlated with its molecular identity. An AI can learn those correlations at a scale that's just impossible for a person.
I can maybe see some obvious ones, like a cluster of inflammatory cells looking a certain way, but it must be finding incredibly subtle patterns.
It is, and to learn those patterns, they needed an absolutely monumental training set.
This is the data from Providence Health.
Yeah. They had 40 million cells with perfectly paired data. So for each of the 40 million cells, they had both the H&E image and the real MIF data.
40 million pairs.
40 million. That's the scale you need for the model to learn the difference between two cells that might look identical to a pathologist, but are totally different on a molecular level.
And the architecture for this is pretty sophisticated. It's not a simple image-to-image thing.
No, they use something called a nested U-Net. It's an encoder-decoder network.
Okay, so break that down for us.
Think of it like this. The model takes a tiny piece of the H&E slide, maybe a 256 pixel square. That's the encoding phase. It basically compresses all that visual information down
Into a sort of mathematical summary.
Exactly. A summary of all the important shapes and textures. It creates a condensed latent feature representation, a kind of biological code.
And then it uses that code to build the output.
That's the decoding stage. It takes that code and reconstructs it, but this time it generates the 21 separate protein channels. It learns that a cell with a very specific, almost invisible nuclear texture is, say, 95% likely to be a CD8 T cell.
That's pattern recognition on a whole different level. It's unlocking information that was always there, just hidden.
Exactly.
So the AI can identify a single cell. But in cancer, we know location is everything. You can't just count the soldiers. You need to know where they are on the battlefield.
And that brings us to the tumor immune microenvironment, or the TIME.
The time.
Right. And that's where GigaTIME really shines. The TIME isn't just a random bunch of cells. It's a rogue ecosystem, a battlefield with soldiers, supply lines, defensive walls. And a drug success or failure depends entirely on the interactions within that ecosystem.
And before this, you could only analyze that battlefield on a few hundred slides at a time.
Because of the cost, yeah. Now, you can run that same complex spatial analysis on thousands of slides that have only ever had that $5 H&E stain. It's a game changer for seeing interactions that were just invisible before.
Did the study show any examples of that? Of insights that were missed before?
It did, and they were profound. For instance, they found that looking at just one protein by itself often isn't enough to predict anything meaningful.
It's the combination that matters.
It's the combination and the location. The AI showed that when you see CD138 cells, those are plasma cells, and CD68 cells, which are macrophages, close together, that specific combination was a much, much stronger predictor of certain outcomes than looking at either one alone.
And why is that something a human would miss?
Because a pathologist might see general inflammation, but they can't quantify the exact ratio and proximity of two specific cell types across thousands of tumors. The AI can. It quantified a biological conversation between those two cells that turned out to be incredibly important.
Okay, so if you're listening to this, the immediate skeptical question has to be, is this real, or is this just incredibly believable noise? Is the AI just hallucinating, plausible-looking data?
A fair question. And the researchers knew they had to answer it with massive validation.
So they didn't just test it on a small sample?
Not at all. They took the trained model and threw it at a huge, completely unseen, real-world data set. We're talking 14,256 patients.
Wow.
From 51 different hospitals, covering 306 different cancer subtypes, a massive cross-section of the real world.
And what came out of that huge scan?
They found 12,234 statistically significant associations between the virtual protein levels and known clinical biomarkers.
1,200 new potential leads.
In a single computational run. It's a stunning amount of discovery.
Can you give us a concrete example of one of those?
Sure. They found a very strong signal that tumors with a specific genetic mutation, KMT2D, also had really high levels of immune cells, CD3 and CD8 T cells.
Which means they might be hot tumors.
Exactly. It immediately suggests that patients with those tumors might be great candidates for immunotherapy. And that wasn't a hypothesis they went in with. It was something the data revealed to them.
But the real acid test is external validation, right? Proving it's not just memorizing quirks from one hospital system.
Absolutely. So they took the model and applied it to a completely different, globally recognized data set. The Cancer Genome Atlas, TCGA.
And what happened?
The results held up. They applied it to over 10,000 patients from the TCGA, and the virtual protein profiles they generated correlated incredibly strongly with what they found in the first cohort.
How strongly? What are the numbers?
The Spearman correlation was 0.88.
That's incredibly high for this kind of complex biological prediction.
It's almost unheard of. It proves the model's biological intelligence can generalize across completely different patient populations and hospital systems. It wasn't just a fluke.
And they also checked the quality, right, to make sure it wasn't just pretty noise.
Yes, they used a bunch of quality control metrics, things like entropy and signal-to-noise ratio, to check the clarity and biological plausibility of the images themselves. It held up much better than older methods, which often just produced random-looking static.
Now, given how powerful this is, we have to talk about the limitation.
Oh, absolutely. And this is a critical caveat. GigaTIME is strictly for research use only.
This is not a diagnostic tool. You cannot use it to make a decision about an individual patient's care.
Not at all. The researchers are very clear about this. If you go to download the model on a site like Hugging Face, you have to sign an agreement specifically prohibiting clinical use. It's for generating hypotheses, for guiding science, not for treating patients.
And for the researchers listening, what kind of setup do you need to actually run this?
You need some serious hardware. We're talking a Linux environment, powerful NVIDIA GPUs like the A100s, and experience with deep learning frameworks like PyTorch. This is bioinformatics infrastructure.
But when you compare that investment to the savings, I mean, let's talk about the economics. This is the punchline.
It really is. Traditional myth. $1,000 a slide, minimum. Days of work. A cohort of maybe a few hundred.
And with GigaTIME.
The material cost drops to basically zero. The computing cost is less than $1 per slide.
A 99% reduction.
At least. And the time goes from days to minutes. And your cohort size explodes from hundreds to, tens of thousands.
That economic shift just it democratizes this level of discovery. Questions that were once only answerable by the biggest labs are now open to many more people.
Right.
So to sort of bring this all together, GigaTIME is this incredible engine for finding needles in a haystack. It finds those rare, important biological clues in huge archives of tissue.
And it does it quickly and cheaply, letting researchers test ideas virtually before committing millions to a clinical trial.
It really feels like the first big tangible step towards that idea of a digital twin or a virtual patient in cancer research.
I think so. It proves that we can infer these incredibly complex, hidden biological states from simple, abundant data. It doesn't replace the wet lab. It augments it. It lets us ask questions we couldn't even afford to ask before.
So here's the final thought to leave you with, and it's all about that economic impact.
Yeah.
This doesn't just lower the barrier for future discovery, it retroactively unlocks the past. Think about the millions of slides and archives from clinical trials that happened 10, 20 years ago.
Slides where they couldn't afford to stain for crucial markers like PD-L1 or CD8.
Exactly. All of that hidden biological information, that treasure trove of patient history that was thought to be lost to time, is suddenly measurable. It's unlockable. You can go back and reanalyze old trials without cutting a single new piece of tissue. The era of the virtual patient isn't coming. It's already loading.